the Th1 immune response. The human body is constantly under siege. It must defend itself from a whole host of bacterial, parasitic, and viral invaders, not to mention rogue cancer cells. The immune system is the primary line of defense in this game of chess, recognizing and ignoring self antigens while vigorously attacking foreign ones. T lymphocytes, the strategists in this game, both direct the responses of the rest of the immune system and play an active role in it. T cells orchestrate the immune response via the production of cytokines, secreted proteins that instruct other cells to behave in specific ways. Scientists have devoted a great deal of time and effort to understand the development and differentiation of T lymphocytes because of both the pivotal role T cells play in directing the immune response and the interesting developmental paradigms T cells represent. T cells may be broadly classified as either helper T cells, that is Th cells or CD4 plus, or cytotoxic T cells, that is Tc cells or CD8. In 1986, Mossman et al. Observed that individual clones of helper T cells could be separated into two classes, depending upon the specific cytokines the cells secrete in response to antigenic stimuli. The Th1 cells primarily produce interferon, IFN-g, and interleukin 2, whereas Th2 cells produce interleukin 4, IL-5, IL-6, IL-10, and interleukin 13. The two helper T cells classes also differ by the type of immune response they produce. While Th1 cells tend to generate responses against intracellular parasites such as bacteria and viruses, Th2 cells produce immune responses against helminths and other extracellular parasites. Interestingly, the cytokines produced by each Th subset tend to both stimulate production of that Th subset and inhibit development of the other Th subset. That is, IFN-g produced by Th1 cell has the dual effect of both stimulating Th1 development and inhibiting Th2 development. The Th2-secreted interleukin 10, however, has the opposite effect. Helper T cells play a central role in directing the immune response. Whether an antigen elicits a Th1 or a Th2 type response has a profound effect on the ability of the body to clear the infection. Interestingly, the cytokines produced by each Th subset tends to both stimulate production of that Th subset and inhibit development of the other Th subset. That is, IFN-g produced by Th1 cells has the dual effect of both stimulating Th1 development and inhibiting Th2 development. The Th2-secreted IL-10, however, has the opposite effect. There is perhaps no more dramatic demonstration of the functional consequence of the diametrically opposing roles of Th1 and Th2 cells than the ability of the inbred mouse strains to respond to infection with Leishmania major, an intracellular parasite. Most inbred mouse strains respond to Leishmania major infection with a Th1-like response, clearing the infection. In contrast, BALB-C strains which respond with a Th2 response are unable to do so and ultimately succumb to infection. It is unclear as to how newly minted helper T cells precursors can be induced to differentiate into Th1 or Th2 cells in vivo. Cytokines clearly play an important role in the development or maintenance of the Th lineages. But there is an ongoing debate about exactly what that role is. Reiner identifies two competing models, instruction and selection. The instruction model holds that native T cells are instructed to differentiate into either Th1 or Th2 cells by virtue of the cytokine environment that surrounds them. That is, if naive T cells find themselves in the presence of IL-12, they will differentiate into Th1 cells, whereas IL-4 will stimulate the Th2 differentiation. The selection model suggests that the cells randomly choose a differentiation path and the cytokines maintain and select for T cell subsets once they have already committed to a particular fate. Let us have an overview again of Th1 and Th2 responses. What are they? Cytokines are the hormonal messengers responsible for most of the biological effects in the immune system, such as cell mediated immunity and allergy type responses. Although there are numerous cytokines can be functionally divided into two groups: those that are pro inflammatory and those that are essentially anti inflammatory but that promote allergic responses. T lymphocytes are the major source of cytokines. Now, these cells bear antigen specific receptors on their cell surface to allow recognition of foreign pathogens. They can also recognize normal tissue during episodes of autoimmune diseases. There are two main subtypes of T lymphocytes, distinguished by the presence of cell surface molecules known as CD4 and CD8. T lymphocytes expressing CD4 are also known as helper T cells, and these are regarded as being the most prolific cytokine producers. This subset can be further subdivided into Th1 and Th2, and the cytokines they produce are known as Th1 type cytokine and Th2 type cytokines. Th1 type cytokines tend to produce the pro inflammatory responses responsible for killing intracellular parasites and for perpetuating autoimmune responses. Interferon gamma is the main Th1 cytokine. Excessive pro inflammatory responses can lead to uncontrolled tissue damage, so there needs to be a mechanism to counteract this. The Th2 type cytokines include interleukins 4, 5, and 13, which are associated with the promotion of IgE and eosinophilic responses in atopy, and also interleukin 10, which has more of an anti inflammatory response. In excess, Th2 responses will counteract the Th1 mediated microbicidal action. The optimal scenario would be, therefore, to be wherein humans should produce a well balanced Th1 and Th2 responses suited to the immune challenge. An additional strategy is being used to prevent the onset of disease. Now, this involves the study of pregnancy and early postnatal life. Both of these states are chiefly viewed as Th2 phenomenon to reduce the risk of miscarriage. A strong Th2 response is necessary to modify the Th1 cellular response in utero. The fetus can then switch on an immune response early in pregnancy, and because pregnancy is chiefly a Th2 situation, babies tend to be born with a Th2 biased immune responses. These can be switched off rapidly postnatally under the influence of microbiological exposure or can be enhanced by early exposure to allergens. It is also hypothesized that those who go on to develop full-blown allergies may be those who are born with a generally weaker Th1 response, although it is now apparent that babies with allergies produce weak Th1 and Th2 responses. Some people have suggested that immunization programs are responsible for the increasing incidence of atopy. There is, however, no evidence that immunization causes atopy. However, this is not an argument that we should be exposing children to potentially fatal diseases again. If experiencing native diseases reduces the incidence of atopy, then the risk of immunologists must be to develop vaccines that mimic the positive effect of infection. Herein we have a comprehensive review of Th1 and Th2 immune responses.